Hello, and welcome back to The Iceberg, where we delve a bit deeper into the wonders of neuroscience and the world of neurobiology. I'm Shavam Kale, your host, and today we're going to talk about something I find to be quite fascinating: optogenetics. Optogenetics is a technique that, unsurprisingly, combines both optics and genetics, and in a way that is truly fascinating, especially in the field of neurobiology. The main technique optogenetics employs is the use of a type of virus, called adenoviruses, to encode photosensitive protein. Basically, a quick speed breakdown, but viruses are capable of making proteins using viral RNA to do so. The process of doing so is incredibly complicated and probably should be saved for another day, but what you do need to know is that these viruses can create proteins, which are basically capable of 90% of all the functions in your cells. Now, these adenoviruses make photosensitive proteins, which, as it sounds, are proteins that react based on the presence of light, which means that light can be used to control the activation and deactivation of these proteins, which can inhibit or activate other parts of the brain as well. Because this is almost completely non-invasive, since the only thing we are invading the brain with are these harmless viruses, and the fact that these are incredibly precise, targeting specific neurons, optogenetics has been gaining immense popularity in the scientific community. So how does optogenetics even work? Well, we have to consider what we know about neurons. Neurons are a type of cell that communicate through electrical and chemical signals, and they use ion channels to ionize their bodies and shoot an electrical pulse that activates other chemical messengers, which triggers yet another electrical pulse, and so on and so forth, where you cascade to these massive neural networks, and they use these electrical signals to do their desired task, where it could be moving a muscle, thinking about food, even just doing anything as a whole requires these electrical signals. So these photosensitive proteins are basically capable of controlling the specific neuronal signals within these massive networks in your brain, and they can either shut down the pulse or empower it. And that's what scientists really love about optogenetics. Let's assume hypothetically that there are some neurons in your brain right now that contribute to a disease. For example, consider some neurons in your motor cortex which are just completely malfunctional and cause your arms to constantly windmill without just stopping. What optogenetics can do is that these scientists can embed these photosensitive proteins within those specific neurons in your motor cortex that are causing you to windmill constantly. And in this case, the neurons that are causing these windmill patterns, with a mere shine of light, the signal will get interrupted, and then the arms will return back to normal. Now, of course, this is a very, 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 very oversimplified explanation for something incredibly complex, but it's the basic mechanics of how optogenetics can contribute to the field of neurobiology, and the potential that comes with the ability to control the signaling behavior of specific neurons. Most research on optogenetics has actually entered into clinical trial stages, which means they're now going to be testing it on actual patients with neurological disorders, which by itself is a massive, massive step for the presence of optogenetics in the medical field as a whole. Now, some interesting results in the preclinical trials should be discussed as well, because looking at them, you can be really amazed by how incredibly important and powerful these optogenetic procedures can be. For example, in one research paper on optogenetics in mice, a group of researchers implanted light-sensitive proteins within the hippocampus of these mice. And the hippocampus, by the way, is basically the main producer of memories in your brain. It's important in how you're able to remember everything you know. And what they did find was that when you expose these mice to light, the mice were actually able to activate the memories of themselves. And without the light, the mice would lose those memories. Basically, the connection between memories and processing that most Alzheimer patients don't have are effectively connected with the usage of optogenetics and light-sensitive treatments. Further developments into optogenetics may yield some fascinating results pertaining to potential Alzheimer's cures, though we should recognize that most of these results are only applicable to mice and are still not actually tested with real human patients yet, so it's not completely accurate to determine that these are the exact one-to-one replicas that we can do with humans as well. Even in Parkinson's disease, another neurodegenerative disease, we see a potential treatment with the usage of optogenetics, something that many researchers have been trying to focus on in terms of fixing neurodegenerative diseases, specifically the ones with motor dysfunctions, like Parkinson's. A research dedicated to deep brain stimulation, which we have talked about before in a previous Parkinsonian video, with the usage of photogenic triggers actually showed some very significant alleviations in the symptoms of Parkinson's in mice, and another research on deep brain stimulation with optogenetics, though more focused in the axons of the subthalamic nucleus, which is basically a hub for inhibitory movements, which you want to have if you were going to have constant tremors or motor dysfunctions, was actually able to significantly treat Parkinson's disease, and especially the motor deficits. So what is this saying? Well, basically, Parkinsonian disease treatments could be done with optogenetics by saying, well, if we target the motor cortex, where you're getting all these tremors and body movements from which are just uncontrollable, you target that subthalamic nucleus, the axon. Since we're able to do that, we can actually force them to inhibit these unnecessary and constant tremors and prevent this from actually occurring. And lastly, even in the field of vision, I know, it's a funny pun, optogenetics are showing some fascinating results, especially pertaining to color blindness. Researchers basically have found a way to sensitize the cells to light by encoding proteins that further encode light-activated channels and pumps. This basically means that by using the adenoviruses that we mentioned in the beginning, scientists are effectively making proteins in the eyes more sensitive to light, and making sure they gain different functions as a result of this photosensitive activation, which is how they're basically able to alleviate color blindness. There is some research on treating blindness as a whole, but that is still in the works, especially since one of the biggest steps in that field has only managed to get pure blind mice to only partially be able to see, which is still, by the way, a massive step in terms of vision, but not one big enough to apply to actually humans yet, since we're still doing this for mice and we haven't even gotten to finish it with the mice. But with the advent of optogenetics and further research being done in that field, we could actually see a change in that soon. Anyways, I hope you all learned about the importance in this blooming field of optogenetics and how it can define the future of medicine one day. This is Shivam Kale, and as always, keep diving. Peace out.